Good morning friends. My topic is structure of protein. There is a many classification of protein, but this classification which is based on the structure. Okay. And my name is Dhruti from University. Our objective in today's session, we will include the primary structure of protein, secondary structure of protein, tertiary structure of protein and quaternary structure of protein. Let's see the overall introduction of the primary, secondary, tertiary and quaternary structures. In the primary structures, which include simply the amino acid sequence. In the secondary structure of protein describe the how segment of the peptide backbone orient into the regular pattern. Tertiary structure which describe the how the entire protein molecules coiled into the three-dimensional shape. And in the quaternary structure of protein describe how different protein molecules come together to yield a large aggregate molecules. Let's see one by one. This is the level of protein structure. In the first amino acid sequence, secondary structure, tertiary structure, and this is quaternary structure of protein. Right. First, primary structure of protein that describe the order of amino acid sequence that is joined together in a large sequence. Right, you can see here the figure. There is a large amino acid sequence that make the protein. Right. If you in this sequence, if you write the molecular diagram, if you write the chemical formula, right, and if you replace the R. Then it make the real group would you have the primary structure of protein, right? The primary structure of protein, in this you can see there is an abbreviation, right? There is a 3-letter code, there is a 1-letter code also, right? And in the amino acid sequence, there is an N terminal on the left hand side of the diagram and C terminal that is present into the right hand side diagram. You can see here, right? There is a left hand side where the ammonia is present. This is known as the N terminal, and where the carboxyl group is present, known as the C terminal. Right. For example, there is a sulfur bridge. Right. For example, in the amino acid sequence, there is a 2 cysteine. Right. And the cysteine, there is a 1 SH group is present. Right. If the 2 cysteine side chain end up into the next to each other because of the folding into the polypeptide chain, they can react and they form the sulfur bridges. You can see here there is a 2 cysteine is present, right, and it's converted into the sulfur bridge that is formed into the primary structure of protein. Right. Next we will see the secondary structure of protein. There is a diagram of the secondary structure of protein. In the secondary structure of protein, we will discuss about the 2 type alpha helix and beta pleated sheet. Right. Conformation of the polypeptide chain. There is a 1 amino acid sequence. It is also known as 1 polypeptide chain. Right. So twisting or folding of the polypeptide chain that is referred to a secondary structure of protein. Right. And there is a 2 type of the secondary structure, alpha helix and beta sheet. Right. First we will see the alpha helix. Right. That is the most common structure of the protein. That is the rigid arrangement of the polypeptide chain and the silent features of the alpha helix. In the silent features, the first one is the alpha helix is a tightly coiled packed structure with the amino acid chain extending outward from the central axis. Right, you can see here the central. If you imagine this is a central axis, right? That is the outward chain extending from the central axis. Right. And in the alpha helix, that is stabilized by the hydrogen bond that is formed. For example, there is a 1 polypeptide chain, right? There is you can see here in the diagram. This is the amino acid sequence. This is alpha helix, right? Here you can see this is the carboxylic groups COO and NH, right? So this is a hydrogen bonding that is formed, oxygen from the CO group and hydrogen from the NH group. and this hydrogen bonding that is stabilize the alpha helix, right. A hydrogen bond is a weak, but a hydrogen bond is strong enough to stabilize the alpha helix, right. All the peptide bond except the first and last in the polypeptide chain participate into the hydrogen bonding, right. Each turn of the alpha helix, for example, each turn that include the 3.6 amino acid, right, and the distance between this amino acid, that is the 0.54 nanometer, and the distance you can see here in the figure. This is the one turn of the alpha helix, right, and this one turn that means the 0.54 nanometer per turn, right, and which include the 3.6 amino acid per turn, right, and the distance and the spacing of the each amino acid, that is 0.15 nanometer. This alpha helix, which is stable conformation spontaneously with the lowest energy, right. In the alpha helix there is a two division, right-handed alpha helix and left-handed alpha helix. We will see the how can we identify the right-handed alpha helix and left-handed alpha helix, right. How to measure, right. So take your right hand, right. This is my right hand, right, and the right hand fist your hand, right. I'll fist your hand. For example, this is my thumb, right. This is my thumb, and alpha helix. Think of the alpha helix spiraling of your right thumb in the direction, right. For example, this is my thumb, right, and my finger which goes on to the counterclockwise direction, right. So this is your alpha helix, and this is alpha helix, right, and my fingers which goes on to the counterclockwise, right. You can see here the figure, right, and this side, this is a alpha helix, that is the counterclockwise, right, and this is my left hand, right, and my finger which is curled into the clockwise direction, right. So right hand alpha helix, the direction is counterclockwise, and on the left hand there is a clockwise direction. Next one is the beta pleated sheet, right, and in the beta pleated sheet the hydrogen bond also formed between the neighboring segment of the polypeptide chain. There is a two type, parallel and anti-parallel of the beta sheet, right. You can see here the parallel and anti-parallel, right. This is this B1 figure B, that is the parallel, and this is anti-parallel. Why this is the parallel? Because they have, for example, there is 3 polypeptide chain, right, and this 3 polypeptide chain which goes from N terminal to C. This is the first polypeptide chain, N terminal to C. Second, N terminal to C, and third one, this is N terminal to C terminal, right. So this is a parallel, and in the anti-parallel there is a 2 polypeptide chain. The one polypeptide, there is a N terminal to C terminal, right, and in the second polypeptide chain there is a N terminal to C terminal, right. Means in the first polypeptide chain, N terminal, below the N terminal there is a C terminal, and in the right in the left side there is a C terminal, and in the C terminal below the N terminal, right. You can see here this is the anti-parallel, and this is the parallel. Next one, we will see the tertiary structure of protein, right. Tertiary structure of protein determined by a variety of bonding interaction between a side chain on the amino acids, and this bonding interaction may be stronger compared than the hydrogen bonding, right, and bonding interaction between the side chain which is depend on to the number of folds, bands and loops that is present into the protein chain, right. Bonding interaction between the side chain may cause a number of fold, bands and loops into the protein chain. There is a different fragment of the some chain may become bonded together. There is a 4 type of the bonding. First is hydrogen bonding, second one salt bridge, third one disulfide bond, and the fourth one non-polar hydrophobic interaction. This is the example of the disulfide. cysteine which contain the one SH and another cysteine which contain the another SH, right. You can see here there is a disulfide bond is present, oxidation of the sulfhydryl group that is present into the cysteine, right, and the two molecule of the cysteine which make the cysteine. Hydrogen bonding that is also formed between the two alcohol, between the alcohol and acid, between two acid and between the alcohol and amide or amine group. Let's see the first example of the hydrogen bonding, right, and this is the asparagine and this is tyrosine, right. Tyrosine OH group and asparagine carboxylic oxygen, this hydrogen and oxygen in this between two amino acid there is a hydrogen bond is formed, right. This is the example of the hydrogen bonding. Next one is the salt wedge. Salt that means for example anion and cation, right. Salt wedge that means the combination of the anion and cation. Bonding between the anion and cation, anion that is the negatively charged and cation that is the positively charged ions, right. Neutralization of acid and amide on the side chain and the interaction that is ionic, right, because the interaction which is occurred between the ions, between the positive and negative ion. The positive ammonium group and the negative carboxylic or we can say acidic group, right. You can see here the example glutamic acid that is the negative or anion and lysine that is the positive, right. So bonding between this glutamic acid and lysine that is the salt wedge. Why? Because the interaction between the cation and anion, right. Non-polar hydrophobic interaction. Non-polar hydrophobic that means water hating and hydrophilic that means water loving. Hydrophobic interaction between the non-polar side chain which are believed to contribute significantly to stabilize the tertiary structure in proteins. Non-polar group mutually repel the water, right, and each other and the other groups results in the net interaction of the non-polar group for the each other, right. You can see here the example hydrophobic interaction between the leucine and phenylalanine, right. In the leucine and that is also non-polar, right, and another leucine molecules that is also non-polar, right. So this is a non-polar hydrophobic interaction. Next one is the quaternary structure of protein, and the quaternary structure of protein which involves several individual peptide or protein chain into a final specific shape. There is a two major category of the quaternary structure of protein, fibrous and globular, right. The fibrous protein, the final beta shaped structure of the silk that is the example of silk which contain the fibrous protein, right. From the diagram you can identify the clear difference between the fibrous and globular protein, right. Fibrous protein, example of the fibrous protein that is collagen. There is a simple polypeptide that is in the rod shape, right, and this globular that means the polypeptide which is clumped together and form a ball shape, right. Example of fibrous protein that is also present into the keratin in the wool and hair which is also composed of the hair. In the hair keratin protein is present, and this keratin which contain the which compose the alpha helical protein chain, right. This is the quaternary protein structure: collagen, silk and insulin. Collagen and silk that is the example of the fibrous protein, and insulin that is the example of the globular protein, right. The best example of the globular protein that is the hemoglobin, right. Globular protein may have a combination of the above type of these structures and mostly clumped into the shape of the ball, right. For example, insulin, hemoglobin and most enzymes. This is the collagen structure. Human insulin which contain the two polypeptide chain with a total 51 amino acids. This chain which is connected together by the two disulfide bond. Insulin that is also classified as a hormones and that is also needed for the diabetic patients, right, for the utilization of the glucose, right. And human insulin that is manufactured from the recombinant DNA technology, right. Human insulin are also low, and it is also obtained from the cows, hogs and sheep. The secondary structure of the insulin, that is the alpha helix, right, and intramolecular hydrogen bonding in the alpha helix is between the amide group. The hydrogen on the amide of the one protein chain is hydrogen bonded to the another amide oxygen on the neighboring protein chain. Thank you.